Good morning, everyone, and welcome to our Food Industry Development webinar. So, broadcasting here from Teagasc Ashtown, my name is Carol Griffin, and I work with the Food Industry Development Department. And today we're going to have a look at what our Food Imaging Centre based in Teagasc and Moore Park does. So Laura Mascaraque and Deirdre Kennedy will be talking to you for about 25 minutes. And please feel free to put your questions into the Q&A function, and we'll have a lovely session at the end. So we'll ask the girls questions, and it's a very interesting topic. So as you go along, think of the questions and add them, and we'll be having about 15 or 20 minutes at the end. So food industry development here in Ashtown and Moore Park, client facing, so we would work in services to businesses. So just to give you a little more about Deirdre and Laura. So Deirdre is a technologist and she manages the Food Imaging Centre in Moore Park, the client facing side. And Laura is a research officer based in the same centre, and she looks at new and emerging microscopy techniques and how they can be used to assess microstructure. So I'm going to, please, this webinar is being recorded. So if it all shuts down on you or you lose connection, it is being recorded. So we will send out the links to that. And if we don't get to all the questions today, Laura and Deirdre will be giving their email addresses, but also we will answer and send out directly on email to participants. So I'm going to hand you over now to Deirdre. So enjoy the webinar. Thanks, Deirdre.
Thank you, Carol, and thanks for your introduction and the opportunity to present here today. So as Carol has said, we both collectively run the food microstructure laboratories in Teagasc. I tend to be the main point of contact for clients looking for help with the product analysis, and Laura later on today is the lead researcher in the area. So for today, we ask that if you have any questions, you can just, again, as Carol said, pop them into the question and answer box. And if you need to make contact with us at any point after the seminar, our contact emails are here on this next slide here, Carol. Great, okay. So again, as Carol has said, we are based in Teagasc Moore Park in Fermoy. So we're actually in the food chemistry and technology department. So the main focus of our department is this dairy based research. We have many analytical laboratories, food structure laboratories, and pilot plant facilities. So we collectively term our specific laboratories the National Food Imaging Centre, where the specific area of food microscopy is our focus. So we can see here on the slide just some of the main tools that we use to characterize food structures. So I just wanted to start by saying, you know, some of the large and leading food producers with these extensive research and development capabilities, these guys know that focusing on food microstructure can benefit their commercial agendas. So some of these big players might have the similar types of facilities that we have. We feel that the small to medium food producers can benefit from accessing both the equipment and the expertise here in the Food Imaging Centre. It's often complemented by collaboration with our colleagues in our department and other departments within Moore Park and Ashtown campuses. So I just want to start off by introducing the concept of food structure. So firstly, we can say that all foods have a structure, that this structure is formed on possibly different levels by a combination of the various ingredients and processes that it is subjected to. For example, if something damaged the structure of our house, it would have major repercussions for the entire house and how it functions. So we also need to think of foods in the same way. We need to understand how processing of foods create the product architecture and how ingredients and raw materials interact to form these structures. So knowing the formulations is just not enough. For example, the same ingredients that are used to make cream, where water is the continuous phase, are also used to make butter, which is an oil continuous spread. So microscopy allows us to examine and see structures that the naked eye can't resolve. And this is exactly what we do. So different food components and different food structures vary in size, and different techniques may be needed to look at them. So for example, an emulsion might contain droplets that are often at the mesoscale, and they also need to be a certain size in order for an emulsion to remain stable. At the nanoscale, we might be looking at individual proteins that maybe under certain conditions become a larger structure, such as a gel. So an example of this would be milk becoming yogurt. So just to really say that food systems, they are complex, and the structure, it can be very complex, and its properties are determined by the physical and chemical properties of the individual components and their interactions. So the structure is essentially the matrix that's responsible for nutrient delivery and functionality. So it affects how food needs to be packed, how food needs to be stored, and very importantly plays a big role in how it's perceived by consumers, both visually and orally, an example being the perceived texture or mouthfeel. So there is evidence that most textural attributes of foods are perceived through mastication, so when we chew them in our mouths. So interestingly, in a study, people were given different foods to eat blindfolded. So they could only identify less than half of the foods when these had been pureed, which destroyed the original structure of the food products. So composition analysis, while it's very important and something that we routinely do, it's not enough to define foods. So the structure is also key here. So structural information is also needed to understand nutritional, functional, and stability properties of foods also. So using microscopy, as we have mentioned, we can visualize components at the micro and nano scale. We can identify components as some structures may have a defined morphology, such as sugar crystals, for example. Where something is located in a product might be of interest. Is it distributed homogeneously throughout a product? For example, if you ended up with a layer of sediment in your beverage, we might investigate what this component is and if it could be better distributed throughout the same product by maybe improving homogenization, or maybe we need to choose a different stabilizer. So modifying processes or ingredients can drastically change the structure of a food due to these interactions that can take place. So why would companies use our service? So we aim to provide this scientific support to businesses, and we can help at the ways listed here on this slide. So the facilities and in-house knowledge of food structure could provide a lot of information to food businesses to help with understanding products and processes, in troubleshooting some issues, maybe to help with shelf life analysis or predictions. The service also can provide visuals that might be needed in the context of other analysis. So these visuals can be very well communicated to others, such as stakeholders, clients, management, marketing people, and so on. We find that clients always welcome these visuals for their own presentations. So just to give one example, on this slide, we can see an example of an experimental design or analysis that might be performed on a new yogurt product as it's being developed. So just to say that the microstructure is only one element to the overall analysis that we might recommend. So the results of all the types of tests are often linked to one another. For example, if we were to do a rheological test, we might measure how strong the gel is. So a set yogurt, for example, would have a different gel strength to a drinkable yogurt. This would also be reflected in the imaging. So if the formulation was to change, for example, if you lowered the fat content, this would be evident straight away in the microstructure, and also likely to be reflected in all the other analysis also. So these other types of structural and sensory analysis can also be performed with our other colleagues here in Teagasc, thus giving us a full picture of what is going on within food products. So I won't go through all the examples of equipment and type of test that we do. I just want to have a look at a few and concentrate on a few examples. So the first type of analysis I'll mention briefly is light microscopy. So a very, very simple technique can tell us a lot about food structures. So morphological information, such as the size and shape of starch granules, can actually allow us to identify the origin of the starch, as potato starch has a different morphology to wheat starch, for example. We might be able to use stains to identify components and tell us more about the structure of a food. Something like sugars crystallizing in a honey sample and incompatible phases in a liquid sample can be seen using specific filters on a microscope. So this might allude to things like your storage conditions being incorrect, or maybe a different stabilizer being needed in your product. So just an example in this slide, we can illustrate how a single food structure might change in the presence of heating. So when starch is combined with heat and water, individual starch granules absorb liquid and swell. So this process is called gelatinisation, and we might rely upon it to cause liquid to thicken, for example, in sauces. So as you can see here, we can actually image at various temperatures, controlling the temperature on a stage under the microscope, and see when the starch begins to swell and how the structure change occurs. So this is just one example of how we might investigate what happens to a structure as a result of cooking or further processing, and may be able to give information on the type of processes that might be needed. So the next useful technique for looking at food structure is our Confocal laser scanning microscope. So this equipment uses fluorescent probes to label different food components, and then we can try and visualize the overall structure of the food. So a common food emulsion, mayonnaise, is shown here on this slide. So you can see that it's composed of droplets of vegetable oil, shown here in green, and these are surrounded by proteins in red, likely to be from the egg yolk, which is used as an emulsifier. So we can see that it is present at the interface where we would expect it to be. So if an emulsifier wasn't doing its job, maybe we wouldn't see it present at the components interfaces, and we might have to address this in a product. So another example here is using the same technique, how we can examine the structure of bread. So if you were to change the type of grain that you were using, or the protein source, or you were reformulating to make gluten-free bread or so on, this change in microstructure could also be detailed and interpreted to help with this. So I just wanted to show how various types of the same products actually have completely different structures. So if we look at the mozzarella, we can see mozzarella itself is subjected to cooking and stretching, and you can see this elongated structures, which is related to its stringiness. You can compare this to a cheddar, as you can see just right above it. So we can also note a high fat content in the cream cheese sample, and the processed cheese sample may have more of a homogeneous network as it had additional steps during its production. So for cheese, these types of images might help us understand their functionalities when it comes to things like melting. So we can use another kind of microscope, electron microscopy, to look at sample in more detail as it has higher resolution. So the microscope can be useful for looking at morphology and surface topography of samples, such as the powders shown here. So we can see the level of detail we can get using electron microscopy, seen here on the bottom row, versus a standard optical microscope, which is the top row here on the slide. So this is very useful for looking at things of how the surface of a powder might change over time. Here you have again the example of the milk powder stored at different relative humidity conditions. So microscopy shows us that in the sample stored at high relative humidities on the right-hand side of the screen, lactose has actually crystallized within the powder, and these powders are now likely to have altered physical properties. So this has implications for shelf life, storage conditions, and functionality of the product. So foods are not often suitable by this conventional analysis by SEM because they are complex multi-phase systems, and they often can contain high amounts of waters and lipids and so on. So we actually have the ability to image under cryogenic conditions, which is where we use specialized equipment to freeze and image samples using liquid nitrogen. So the liquid nitrogen preserves them in their natural state, and they're held at these low temperatures while being imaged. So the Cryo SEM allows us again to see high levels of detail. As shown here, we see whipped cream, we can see air bubbles and the aqueous phase containing the fat, showing its air and water emulsion. Also, we can fracture a sample in its frozen state to reveal a structure below the surface to give us even more information. So on the bottom of the slide here, we can see that we can actually resolve individual bacteria in a yogurt or individual fat droplets in an already tiny powder particle. So I just want to take a look at this point, how we might approach a product problem for a customer. So just going to use the example here of a dairy-based coffee creamer. So this is a type of instant powder that may be sold as an ingredient or as an end product. So for this type of product to be successful, it would be expected to impart a creamy taste and a whiter color to beverages. It would need to be agglomerated, i.e. instantized. It needs to not clump when it goes into the liquid. It very importantly needs to not precipitate or fleck when added to a coffee. And it also needs to pass a sedimentation test. So if a customer was having issues or wanted quality check powders, apart from the routine standard coffee tests and sedimentation tests, which we can also do, we might look at them under the various microscopes. So in this slide, we see the light and electron microscopes. For this, we might use them to see if the agglomeration was sufficient, to see the size of the powders, how porous they are, as all these things might affect how they would further solubilize. So in this slide, we can see how the labeling technique might identify some issues in such a product. So on the left side, we see a powder that has non-homogenized free fat. So this might cause problems such as oxidation, and powders would likely to have poor wettability, which means they won't initially take up water well. On the right-hand side, we see a powder which is actually showing some protein aggregation. So these powders may solubilize poorly or cause this flecking upon rehydration. We can also help identify differences between batches and how they rehydrate using these high-speed camera type techniques shown on the bottom of the slide. So we can actually image and create videos of how two types of the same powder that perform differently when they are rehydrated. So in this example, one powder solubilizes better than the other, and we can actually prove this. So again, following on from this, some of us may be familiar with the issue of when a powder doesn't solubilize fully, it might leave these little flecks in our coffee or feather or foam, or maybe on the side of a baby's bottle, we see these little flecks, as we call them. So there's many reasons why a powder might have these solubility issues, and there's a huge science behind it. So what we might be able to do is firstly identify what these flecks are composed of when we isolate them and look at them under the microscope. So on the left-hand side, and we have seen in the past, a lot of flecks show up as completely insoluble intact powder particles. We have seen flecks turning up as entirely composed entirely of protein and entirely of lipid. So if we can find out this information for researchers or for manufacturers, they then might consider some reverse engineering that they need to do in order to solve their product problems. So that's it from me. At this stage, I'm going to pass over to Lara, who's going to talk through some more of the techniques.
Thanks, Deirdre, and good morning to everyone. I'd like to very briefly present another microscopy technique that has enormous potential for product troubleshooting for food businesses, and it's called Confocal Raman microscopy. And its main advantage is that it can provide not only structural information, but also chemical or compositional information without the need of staining the samples that sometimes can alter them. For every single pixel of the image, this technique gives us a Raman spectrum, which is will look something like the ones shown on the right image with different peaks. And these peaks contain invaluable chemical information because the Raman spectra are different for every compound, and they are like the fingerprint of each different component. This can give us additional information of the samples that other microscopy techniques can't provide. This information can be extremely useful when it comes to developing new food products. I'll explain it with this practical example. These Raman images were obtained as part of a research piece in which we were investigating how different processing conditions and ingredient formulation affected the nutritional properties and stability of a beverage, in which we had incorporated beta-carotene, which is a provitamin. On the left, we can see the distribution of fat droplets in red and protein in green, obtained through one of the techniques that Deirdre explained before, confocal laser scanning microscopy. But on the figures on the right, obtained by Raman microscopy, we can see much more than that. We can see the adipose phase that contains the protein in blue, the fat droplets in yellow, and the provitamin in red. And the interesting part is that depending on the formulation we used, the provitamin was distributed homogeneously among all the fat droplets, as we can see in the image at the bottom, or selectively in some of them, like in the image at the top. And this will have implications for the way this provitamin, which is usually poorly absorbed in the intestine, will be assimilated by our body. So this type of information, tracking a very specific micronutrient like this provitamin, can't be obtained with the other microscopy techniques we've talked about previously. And another area in which Teagasc research and expertise can be used by food businesses is in building and studying novel food structures. Thanks to the different techniques we've mentioned before, we can help clients obtain very useful information to assist in their new product development and product reformulation processes. Consumers are increasingly demanding products which contain plant proteins. And this example compares the structure of some protein gels produced with either dairy or plant proteins or a mixture of both. So the three gels were analyzed by both by electronic microscopy, which are the gray images, and confocal microscopy, which are the green images. And the first structure of on the left corresponds to a protein gel prepared with soy protein. And the one on the right was prepared with whey proteins. And the one in the middle is the mixture of both, where we can observe how new interesting structures are formed. The different structures of the gels can be correlated with their mechanical properties, which are related to the functional attributes such as mouthfeel, for example. So studying the structure of the products, we can get information of the impact that replacing milk proteins by plant proteins, for example, can have on our products. Both electronic and confocal microscopy have different advantages. In this case, electronic microscopy, the gray images, has a much better resolution, and we can observe details that we can't see with other techniques. But confocal microscopy can also show the main structural features in a most cost-effective way, because the analysis is faster and requires fewer resources. And then in the corner, we have an image obtained for one of them using Raman microscopy that gives us additional information. Apart from the protein structure, we can also detect other components that we were not even looking for. See the green bits there? That turn out to be residual fats that the protein ingredients contained. And with Raman microscopy, because we have all the chemical information of all the pixels, we can potentially detect contamination with other ingredients as well. And finally, the ultimate goal of food is to be consumed and digested to provide nutrients. So in collaboration with other groups in Teagasc who are specialized in food digestion, we also study the changes that these products experience during gastrointestinal digestion. Usually we simulate the digestion in vitro and study how the food products are broken down. So here, for example, the different protein gels that have different microstructures are broken down differently during digestion. And this means that even if the protein content in both is the same, they could be assimilated differently, and they could each have a different satiating effect, for example, as they will stay different times in our stomach. And of course, we will then need to use complementary techniques to actually assess how the proteins are broken down at a molecular level to know which one will absorb better in the intestine. But microscopy can help explaining why by looking at how the product is disintegrated in the stomach at the microstructural level. So I hope this brief description of some of our work has given you an idea of what we can do for food businesses in the area of food microstructure and how we can assist you with your product development and process troubleshooting. And if you have any questions for us, please feel free to ask them now or contact us anytime through one of these email addresses. Thank you.
That's great, Lyra. Thanks a million. I'm just going to stop sharing for a second. Great, so we can see you there. And okay, brilliant. So we have some questions here. So I'm just going to try and interpret. Some of them are a little bit long, but there's someone here from a company who wishes to bring a dairy product to market. So they know the processes that they want to use, but feel that food imaging would give them more valuable information. It's a fascinating side of food assessment. So how does one go about becoming a client? And I think this is a startup person. So maybe Deirdre, just maybe bring me through how someone might, the usual contact initiation and how client might work with you.
Yeah, sure. Again, you know, each client is different. Each case is different. But once they make contact with us, you know, we decide then what the next step forward is. So the best thing would be to pop me an email, or myself and Lara. If we get requests for each other, we'll pass them on to each other and so on. So the best thing would be to pop me an email, and then maybe we could, you know, arrange to give that person a call and talk them through. We would need to get a feel for what they're looking for from us, and we'd be able to advise them then on what we could do for them. And if they're happy with that, then they might send us some samples or come to us and meet us or so on. And, you know, we'd take it from there. So it really, really does depend on the aspect. Sometimes we have clients in-house. Sometimes we have clients using our pilot plant facilities. And sometimes they might just walk up the hall and need something imaged straight away because it's really, it might help their process on the day. So, it really does depend, but I would say always feel free to contact us and to ask, and we will talk you through every step of the process.
Okay. So would, just in case someone is worried about confidentiality or giving you, I presume giving you information would be essential in order for you to identify what you need to do for them.
Well, it's up to the client. If they don't want to give us too much information and they know kind of what analysis they want and what they might be able to interpret from it, that's fine. We do have some maybe repeat clients that know what they want, or maybe they're able to interpret some of these images themselves because we've done stuff for them in the past. But I would say the more information that we get, the more we can give back. So that is again up to the client on a case-by-case basis. We do have a client confidentiality system. Each case is assigned a unique number, and we don't share any of that information with anybody but ourselves. And we're completely secure from a file point of view and everything like that. So we do aim to offer all that to customers, complete confidentiality.
Okay, that's great, thank you. And I presume there could be some funding there that people can access if they need to. We're linked with a lot of funding agencies.
I don't know. On our end, we tend to have specific ways that we would charge to clients or to projects. I suppose maybe the kind of work that you guys do with regards startups and things like that, I would probably have to refer onto someone else. Yeah.
So it's a collaborative. It might not just be the imaging. You could someone.
Yeah, exactly. It might come through someone in your department or something. We often get people come through Eddie O'Neill or someone like that, so we can liaise with Eddie about the funding or whatever. So always best to just contact him. We take everything on a case-by-case basis, I would say.
Okay, great. And I got one for you, Lara. So if Raman Microscopy can give information about the composition of samples, so how much information will be able to find out about something like how much fat is in a product? It's a fairly specific question.
Yeah, so well, with a Raman microscope, and it's the same with any other microscopy techniques, we can't put a number on the fat content on a product in terms of grams of fat per 100 grams of product. What we can do is to see the distribution of fat in that product and estimate the volumetric ratio. There are other techniques to quantify the mass of fat or any other components, and we collaborate with other teams in Teagasc to do complementary analysis if required. But the main information we can get is structural. So like, is the fat forming dispersed droplets in an emulsion, or is it encapsulated within our dairy powders, or is it in the surface? So, and that can also be done with confocal laser scanning microscopy in a more cost-effective way, actually. But the advantage of Raman microscope is that you can distinguish, for example, between lipids with a higher degree of unsaturation from those with a lower degree of unsaturation. That kind of chemical information, we can even distinguish if at a certain temperature this fat is liquid or solid, because we have the different spectra, which, as I mentioned, are like fingerprints. We have invaluable chemical information, so we can distinguish these type of things that we can't do with other microscopy techniques.
Great, that's lovely, thank you. So how would someone decide what's the best microscopy technique to use, or how would you decide? Maybe Deirdre, bring us through that.
Yeah, I suppose again, we would talk to the client. Like I said, some clients might already have an idea of what they want. A lot of people wouldn't. They would probably, again, we'd talk to them, see what kind of product they have, what they're looking to see. Most importantly, I would ask people, what are you looking to get out of it? Because, people will have something maybe in the back of their minds that, I want to see, did this stabilizer work? Or I want to see that did my homogenization do the job? Or I want to compare it with some product that's already on the market. I want to compare my two batches or something like that. So again, just from experience, and we do have a lot of experts in-house on different types of products. So if I had to refer to somebody else, I might have to. And I would recommend a starting point, maybe one of the more simpler techniques And if usually maybe a simple technique might allude to something that I'd say, well, it's also maybe suitable for another technique. So again, I would probably have to discuss with the client on a case-by-case basis. But I would say that all the different microscopy techniques all do different things and all have advantages and disadvantages. So it would definitely be maybe on a product by product basis that we would decide.
Yeah, so it sounds like you might need to start first and see where it's going to lead you.
Absolutely. And I would always say to customers, send in maybe 1 or 2 samples, and we can image 1 or 2 samples for you. And if you like what you see, then you want to send in your other 10 samples, or so like if you're not sure if it's going to work, or you don't want to heavily invest much time or money or whatever, if you want to have a trial to see, we can see, can we see this component you're looking for, or can we help with, send maybe 1 or 2 samples at first if you're not sure if it's a service that you want to use. And then if you're happy with it, then we can organize more analysis. Some people do that starting out.
Well, that sounds good. So do people, Lara, troubleshooting, say for if there's some components you showed on one of your screens that they found fat that they weren't expecting. Does that happen often, or what kind of things can be found or have been found in your analysis?
Well, we find all kind of different things. Usually when clients come to us, it's because they have an issue. They have a product that is not behaving as it should. And many of the cases, this is related to the microstructure. So as Deirdre said in her presentation, sometimes the fat, instead of being well encapsulated within the powder, is on the surface, and that causes problems of redispersion into solution, or some issue during the processing of the product that resulted in a change in the microstructure, and this has an impact on how the product behaves. So usually they come with us with a problem, and we find what can be the issue in relation to the microstructure of the product.
Okay, that's great, thank you. And one of the slides we saw the beakers with the video was just starting, so dispersing. So you can capture film, Deirdre, as well as images.
Yeah, I suppose that particular technique is just a high-speed camera. So we use it, I suppose, to capture things that I suppose it's too quick for our eye to see. So we can capture up to 100,000 frames per second, and then we can slowly play it back for someone. So something that might happen at a level that your eye can't see, we'd hope to be able to see with something like that. But like very easily, we could capture those videos of just water dissolving in a beaker, and that's a really simple technique to do. And we've used it for clients in the past where they're able to prove, different things about their different batches and so on. So it's often I find it also might be something like something to communicate back to your stakeholders or your marketing people or things like that. So, we can do little videos. We can do, if we have, 3D reconstructions of food analysis, we have software to make little videos out of that and things like that. So again, I would just encourage people to talk to us and see what we can do for them.
And would it be fair to say that the imaging is quite a fast technique versus some other techniques, Lara?
In some cases it is, in some cases it is, in some others, it depends always on the problem that you're trying to solve. So for example, if you are looking at something very at a very nanostructural level, for example, and you have to go for a very specialized microscopy technique, it may be not that fast, but still it may solve your problem. So for example, if we use optical microscopy, confocal microscopy, that's very fast. And it may give you a quick look at your sample. You may see straight away the problem, and then you can reverse engineer your process. But it depends on the problem. So sometimes it's a quick technique. Sometimes it involves like having a collaborative project to investigate in more detail some specific problem that you have.
Okay, and the different departments and colleagues in Teagasc would work together on that.
Definitely, yeah, definitely.
So I was really interested in the digestion. You were saying the provitamin that you could help. Now it's in the stomach, wasn't it? You were looking at how the different proteins, or sorry, the provitamin might be absorbed.
Well, in the case of the provitamin, we didn't do a digestion experiment. We just saw where the provitamin is located within the emulsion. So we had different processing conditions and different formulations. And we saw that in some of them, the provitamin was homogeneously distributed in all the fat droplets, while in others, it seems that the beta carotene was only found in some of the droplets. So that's the kind of structural information we can get, and that will have an impact on the digestion. So we can also do that. We didn't do this in this particular case, but that's something that we can do in collaboration with other colleagues, which are experts in food digestion. And we can see the impact that thing that we saw with microscopy actually has on the absorption of the provitamin.
Great, and a very topical one, Deirdre. You showed slides of the plant protein, the fully whey protein, and the mix of the two. That you could look at the structures of those, or the consequences of adding in non-dairy proteins.
Yeah, I think that was Lara's slide.
Yeah.
Sorry, Lara. Sorry.
Yeah.
Yeah, basically the issue here is not all the proteins are the same. Sometimes you think about the protein content that you want to have in a product because of the nutritional benefits of proteins, but not all proteins are the same. So it's not straightforward to substitute or replace one protein by another. And you could see clearly in those images that by replacing part of the proteins, whey proteins by plant proteins, the structure changes completely, and that will have an impact on the textural properties when you chew the product. Even during digestion, it will break down differently. So it has to be studied how the structure is changed when you replace one type of protein with other, because not all proteins have the same molecular structure, and then they don't aggregate in the same way.
It's really interesting because we'd have a lot of trained sensory panels, mostly with a lot of work done on meat, and then subsequently to be highly trained then to assess non-meat products. So it would be good to have an indication ahead of time what it might be like, because it is quite an expensive thing to assess, to do sensory analysis on absolutely everything. So being able to predict what the texture is likely to be would be really advantageous. It would cut down a lot of time for people. And
yeah, microscopy can give you an idea of this. Yeah.
Yes. Just would there be any products defects, say on the confectionery side, that you might have had an opportunity to look at, Deirdre, or have come through?
I have seen maybe just a couple of cases, something like blooming in chocolate. I think I've looked at that before for Eddie, or maybe some cake type products, which actually again was a similar type of defect was occurring in a like a frozen type product, where we actually saw a similar thing where the sugars and the confectionery inside in the cakes were actually this blooming was starting to occur. So we could kind of just, I suppose, prove that for a customer, and I think they might have had to reformulate differently, maybe with the types of sugars they were using. Or
okay, so that would be a really fast way of identifying the.
Yeah, well, I suppose unfortunately with that, I suppose it's probably the problem has already happened. So I suppose at that point we're helping. So yeah, like it is. Sometimes people might come to us in advance or something, but often we're dealing with the other end of it where there is a problem. So if it is definitely worth a quick look under the microscope is definitely worth, might be able to save some time in looking at these shelf life type studies, or in doing a quick reformulation or something.
Okay, and I think we have time for just one more, just a question. The gelatinization of the granules at different temperatures. That's really interesting to be able to see what's happening at different temperatures. And I presume that will help inform people about the process they should undergo or to change the type of starch that they're using.
Yeah, I guess we've seen, I suppose, again, we probably have cereal scientists that know a lot more about this kind of stuff, but we have seen, you'd have products that would have a certain amount of starch in them. Some people using starch, as thickening agents or bulking agents. So there is a chemistry behind each type of starch, and sometimes people might need to blend the amount of starch because starch has this heating gelatinization profile. They're all individual for the different types of starches. So we might have to, so what we might be able to do is see how, if you've chosen to use a particular type of starch inside your formulation, it might act differently because you have proteins in there that might have this protective effect over it, or different types of proteins would have different types of effects actually on the starches. So what we would maybe be able to help with is, again, if you had your product trials or so on, and you wanted to try different types of starch in your same protein, or you want to try different type of proteins with your same starch, maybe to see how this structure reacts to your processing, or how your final product, I suppose the structure of your final product. And I guarantee there will be differences if you were using different starches or different protein sources and things like that. So it is definitely these case to case and these trial to trial kind of analyses, it can definitely help with that.
That's really interesting that, it's an off the shelf or it's a standard starch, but depending on your ingredients, it's going to act very differently.
Yeah, absolutely. Yeah.
Okay. That's all we have time for. So if you have any more questions, please feel free to email Deirdre and Lara, or send in and we'll answer if you want us to answer directly to you. So thank you all very much. Thank you for the very interesting presentation and for all your questions. And this will be available, so we'll send out information here on the Teagasc website that it'll be available afterwards within a few days. So thank you very much. Goodbye. Thanks, ladies. Bye.
Bye now.